Hey guys, it's Miss Larson here talking about DNA fingerprinting. Now, DNA fingerprinting is not using a literal fingerprint like that. It's actually different. So, I want to talk to you about how that works. Here's some background information. Almost every cell in our body contains DNA, and on average, 99.9% of the DNA between two humans is exactly the same. The remaining percentage is what makes us unique. Unless you're an identical twin, then you have identical DNA to somebody else. But although this might sound like a small amount, it means that there are about 3 million base pairs that are different between two people. These differences can be compared and used to help distinguish you from someone else. Many satellites are short sequences, about 10 to 60 base pairs long, of repetitive DNA that show greater variation from one person to the next. And that's going to be based on other parts of our genome. This variation is exhibited in the number of repeated units or stutters in the mini satellite sequence. The first mini satellite was discovered in 1980. So, how does DNA fingerprinting work? Well, DNA fingerprinting was invented in 1984 by Professor Sir Alec Jeffreys after he realized you could detect variations in human DNA in the form of these mini satellites. DNA fingerprinting is a technique that simultaneously detects a lot of mini satellites in the genome and produces a pattern unique to the individual. That pattern is your DNA fingerprint. The probability of two people with the same DNA fingerprint that are not identical twins is very small. Just like your actual fingerprint, your DNA fingerprint is something you're born with and is unique to you. So, I'm going to explain how this process works. I'm actually going to skip the words and show you the picture, and I'll just explain it with the picture. So, if you want to read the words, you may go back to them, but otherwise, think about this. You're going to have a DNA sample, which comes from blood. At least the old way of doing this, you would get your sample from blood. You would extract the DNA out of the blood, and then you would use these things called restriction enzymes that act like scissors to chop that DNA up. Then you load that chopped up DNA into what's called an electrophoresis gel machine, and you load it in right there. And then you put electrodes on one end and the other, and you apply electricity to this electrophoresis gel. Well, what happens then is that because of the electric current, the restriction where the restriction enzymes cut the DNA into different pieces, the DNA starts to separate. It's kind of like chromatography if you've ever done that. But the shorter DNA fragments end up down here. So, like these ones that are shorter, they would end up over here. And the longer DNA fragments stay close to where they were first put in. So, these longer pieces of DNA would stay up close to where the DNA was initially put in the electrophoresis gel. So, you put the sample in, you electrify it, and the DNA separates, with the shortest pieces going the farthest away and the longest pieces staying shorter. Then you transfer that information onto what's called a membrane, and you label the membrane with probes that are radioactive. And when you label it with radioactive probes, you can then take an x ray of your gel electrophoresis, and that gives you your DNA fingerprint. Now, nowadays, there's a lot more technology for DNA profiling, and it involves things called STR, short tandem repeats, and polymerase chain reactions to produce a lot of copies of your DNA. And so you can get actually a lot better results nowadays, but it's a little bit more complicated of a process. So you would extract the DNA, and then you would label the DNA and chop it up using PCR, or polymerase chain reaction, and then do electrophoresis with these labeled pieces. And you would get visualization usually through a computer. So if you've seen a show like Forensics Files, you might have seen a DNA profile that kind of looks like that. And that's using modern technology. But in any case, you have to take a DNA sample. You have to pull the DNA out of the sample. You have to chop the sample up into smaller pieces, do electrophoresis, and then you get a DNA profile. And how does this use to solve crimes? Well, DNA profiles are really useful in forensics because only a tiny sample of human material is left behind at a crime could be used to sufficiently identify somebody. In the UK, or the United Kingdom, a complete DNA profile consists of 11 STR sequences plus a sex or gender determiner to confirm the profile is from a man or a woman. Now, all new profiles include an additional 5 STR sequences to provide some more consistency across all of Europe. So if you were in Europe and you were committing a crime, this is the type of thing that would happen. You would have this modern DNA profiling done, and they would use they would use 11 STR sequences. In the USA, the FBI recommends 13 STR sequences are tested, which is more than it is in Europe. And many states are increasing the number of STR sequences that are tested to enable more efficient investigations across state borders. So if you commit a crime, they are going to look at 13 little small pieces of your DNA and be able to compare that to other people. And when a match is made between a crime scene profile and an individual profile, that it identifies a possible suspect. A match between a different crime scene profiles indicates a repeat offender at work, and police can use the DNA evidence to support other evidence and help prosecute somebody for a crime. Complete DNA profiles give really reliable matches and may provide strong evidence of a suspect being guilty or innocent of a crime. So, here's how the gel electrophoresis works for crime scenes. So, this is the gel electrophoresis of a sample taken from a crime. And here are two suspects. And you can see there that suspect one does not match because in the crime scene there's a bar from the STR from the short fragment of DNA. There's a bar right there and that matches suspect 2. Also a bar here, hey, that matches suspect 1, but it also matches suspect 2. Another bar here matches both suspects. This bar only matches suspect 2, and then the bottom bar here only matches suspect number 2. So in this case, even though there are some similarities between the crime scene and suspect 1, there are more similarities between the crime scene and suspect 2. So it would be more likely that suspect 2 committed this crime. So how are DNA profiles stored? Well, they can be stored in a database, a national database. And in the United States, that's called CODIS. And that can help store all of the DNA samples from all individuals who have ever given a DNA sample. And that can be used to catch criminal offenders. So it can also be used to link you to your blood relatives. So you get half your DNA from your mother and half from your father. And STRs, which are those short pieces of DNA, they are therefore passed down actually from your parents to the children. DNA profiling can be used to help confirm whether two people are related to one another, and it is commonly used to provide evidence that someone is or is not the biological parent of a child if there's a custody debate. DNA profiling can also be used to identify victims of a crime or major disasters that could happen and help bring separated families back together. DNA profiling has a really high success rate and a very low false positive rate, so it's fairly reliable. And you can see here using the modern DNA technology how you could identify if the child does belong to this particular mother and father. And you can see this child has markers number 5 and 7. And you can see the mom had 5 and the mom had 10. Well, the mom must have passed down number 5 to the child, but not number 10. And remember, that's because of the law of segregation, and the law of independent assortment could mean that she would only pass down that number 5 marker. So she passes 5 to the child. Now look at the father. Father has 7 and 13. Is it possible that this father is the father of the child? Yes, because the child also has marker number 7. So you can see that this child did in fact come from this mother and father, and you can see which DNA markers it inherited from each parent. Thanks for listening. Hope you enjoyed. And if you have more questions, please do ask me. You can send me an email. We can chat about it.